Chromatography is still relevant in the pharmaceutical industry for fast-paced programs that are moving quickly through to the clinic. In some cases, chemistries can't be optimized and chromatography is necessary to remove impurities that are produced or recover materials that are obtained in a very low yield. Chromatography is typically used to remove impurities that are structurally similar to the target molecule, and sometimes they're also used to purify materials that are not crystalline in nature. They could be oils, waxes, or other materials that are not amenable to a solid isolation. Choosing the right stationary phase for separation typically involves looking at the molecules and the types of functional groups that they possess. If they have a lot of polar functional groups, then a normal phase silica chromatography is typically going to be the most cost-effective solution. If the molecules are very similar in terms of the number of hydrocarbons or aromatic rings, then a reverse phase chromatography stationary phase might be more applicable. Some molecules differ in 3D space. They could be diastereomers or stereoisomers of one another, in which case a chiral stationary phase would be more applicable. There are a number of key steps in chromatography process development. The first step is understanding the chemistry, looking at the kinetics of the target product formation as well as the impurities that are difficult to remove. In the second step, you would want to optimize the chemistry conditions in order to improve the purity of the target molecule, but in particular to address the impurities that are difficult to remove and might require chromatography. The next step in chromatography development involves looking at different mobile phases or solvents that can separate the impurities from the target compound. This is followed by some small-scale column chromatography, where you might look at improving the yield, increasing the column loading, or reducing the amount of solvent that's required to elute the target product. The final stages in developing a scalable chromatographic purification involve isolating the compound from the purified fractions. This might involve some concentration, lyophilization, or precipitation crystallization strategies. Lastly, the timelines for purification and isolation of materials at manufacturing scale can be long. Therefore, you have to look at the stability of the materials throughout the chromatographic separation and during extended holding time during fraction analysis and isolation. Purimal has a full range of equipment to scale up chromatographic purification processes, including normal phase chromatography as well as preparative HPLC that might include reverse phase or chiral separation technologies. Purimal has the experience and expertise to scale up chromatographic separations to deliver on programs that are moving quickly towards the clinic and provide materials to the patients that need them.